Welcome everyone to our endocrine grand rounds. It's my privilege to introduce my friend and colleague, Dr. Lou Kai. Dr. Kai received his MD in 1983 and Ph.D. in 1990 from what is now known as Jilin University, which I have visited actually with him. He was trained as a postdoctoral fellow in Department of Pathology at the University of Western Ontario and the Department of Pharmacology and Therapeutics at McGill University. He joined the University of Louisville in 2000, I believe, and is a tenured professor with appointments in the departments of Pediatrics, Radiation Oncology and Pharmacology, and Toxicology. And in 2013, he became the director of the Pediatric Research Institute at the UofL School of Medicine. In his very active and highly successful career, he's mentored dozens of students and physicians who have gone on to productive research careers. His research has been supported by the AHA, JDRF, DOD, and the NIH in three major fields in which he has more than 320 peer-reviewed publications. These areas are the hormesis and adaptive response induced by low-dose radiation and clinical applications, cardiovascular complications of diabetes and obesity, and systemic and reproductive toxicology of environmental exposures to factors such as radiation and heavy metals. And this last topic is the one in which he will speak to us today. And his presentation is entitled Potential Health Effects of Environmental Exposure to Cadmium on the Victim and Their Offspring. Please welcome Dr. Lou Kai.
Thank you very much, Dr. Copper-Vinegars, and thank you for your nice and kind introduction, very detailed. Thanks a lot. And good afternoon, and everybody, and thank you for your come and in person here, and also some in the online and virtual, and I greatly appreciate. And today is my great honor and be here and share my work and regarding the cadmium and potential health issues. And so I do not have the conflict of interest to disclosure, and except the PI for the ADA and support grant. And I would like to divide the following part of my today's talk, and very briefly talk about the environmental cadmium, and because that is a very well-known metal, so very briefly. And we're focused on my cadmium related to the research. And I would like to divide the two model, and actually it's a two part. And one is the exposure for the adults and look for the long-term and impact. And another model is look for the whole life. That means pre-conception, pre-embryo period until weaning, and then also follow the adulthood and adolescence and also adulthood, and then without other challenge. And then what is the next future plans? So my lab and I work on the diabetic complication. So this is the title I last time to present in here in the March, and actually, we work on the zinc and methylcyanin. Methylcyanin is the metal binding proteins. So that is also bring me to interesting for the metal. And we found the metal is not always is good and very important for our body, but some metal is toxic. So that is related to some why I'm interested in the heavy metal toxicity and related to potential health impact. And like Dr. Winker said, I also have the project regarding the low-dose radiation. So that actually is very interesting, and I won't be sharing here and today, and probably next year if we have some and interesting, I can talk about that. And like environmental and pollute and like a determination of the health and the issue. So that is like actually not like 30 years ago. Probably people won't and appreciate that, but right now is more and more evidence already indicates that environmental pollute and like the food and drinking water and all the contamination cause health problems. My group is interesting for the following target, so I will focus on this area, not like everything. And for the CDC and the state by state and the map, and you can see that in 2020 last year, and and showing the obese and prevalence. And our Kentucky, not that lucky, always in the top, and it's not top one, but at least in the belong to top levels. And compare and 10 years ago and other state kind of the difference. And but our state is same and consistent. And for the cancer and prevalence, more interesting. And you see, this is this 20 years window, and they investigate that. And in the 1999, and several states have a high prevalence for the cancer and the new cancer diagnosis each year. And our Kentucky is here, also belong to the top group. But actually, it's after 20 years later, we are not just the group, we are the leading and the number one in the prevalence for the cancer. So what is the situation? And our state also adopt a lot of the program, exercise, eat the healthy food, and also kind of different program education. But why we are not change always like that way? So that probably one clue is probably the environmental and not the change, because we have a pollute and we have tobacco, we have a heavy kind of probably that contaminate and pollution. Actually, Dr. Rooney Bandagar group really work on the environmental green tree. Probably in another 20 years, probably we will change and probably this follow that. This is my hope. So that is something background, very briefly talk about the environmental, how they affect our health. And when we go to the EPA, we look for the environmental pollute, and they have some different route. You can see the affect air, what we breathe in, and the water, what we drink, and also land. So that give you some kind of plants and the vegetable and the food. Probably you got the food from the land to have contaminate for the bad stuff. And if you look for the indicator, they have a top one as the blood cadmium, and that means that cadmium really is in the top, and some warning and for the environmental caused pollute and cause the health related issues. So that's why, and I want to see and what is the cadmium and why I'm interested in cadmium. And the cadmium already listed the top 7 by the ATSDR, that is kind of the look for the chemical substract and disease registry. So that is kind of the toxic and chemicals. So cadmium belongs to top 7s. And the cadmium actually in our body is nothing useful, and we don't need at all for the cadmium, not like other metal, zinc and copper, and even sometimes it causes bad effect, but that is required for our body. Cadmium is not at all. And where we can find got the cadmium. Cadmium is kind of the byproduct when you generate zinc and lead and copper and coal. They have some and cadmium and generate. And they also have some opportunity to occupational exposure for the cadmium. And you can see and the battery, the factory, the battery made by the have the cadmium and some the plant. And this is the slice is made by my Ph.D. student, Dr. Jimmy Young, and I saw this picture is very pretty, and she's very kind to allow me use this slides a long time ago to do the presentation. So still I want to credit to her. And so not just occupational and environmental, and also related to the cadmium pollute, because for some jewelry from the glassware, they have the cadmium plant that cause our daily life. Probably somehow you are got some contaminant in their contact for the environmental. And further, the fertilizer, probably your backyard or some a farm and a park or whatever, some forest, and so probably they have some increased cadmium pollute too. So basically, year by year, you can see that cadmium production used in the still increased, and even already 2020, so that is increased. So what cadmium and if intake in our body, and this actually is very bad. because cadmium stay our body very long, 30 years, 40 years, not that easy to eliminate. And cadmium not just affect one organ, like diabetes, affect all the organs, and the bone, and the kidney, and the heart, and the lung, and so that is really bad. And even like before last week, and when I go to online to search, and the recent published humans like study, they like kind of the big number, and they look for the cadmium in the urine and in the blood, and also compare, and what they found, and they found is that around the industrial or like smoke area, and got a cadmium intake or inhalation, and go through the lung, and really cause the lung cancer and increased. So that is really something related to the cancer or like some disease we are work on. So my lab, like I mentioned, and I'm interested in diabetes, obesity caused complication, and cardiovascular disease and cancer. So at the same time, I'm thinking diabetic patient is not pure in the world without any contaminant. Actually, it's probably also smoke and environmental and health and cadmium contaminant, and what that's going on, probably that are synergic in fact for this disease. So that is what we work on in the lab. So now I want to detail and introduce my the model 1 and the adulthood and got exposure to see what's going on for the adults. And about 10 years ago, and one pathology and in China, and he come to my lab and and want to work on some project and with me. And his background is a medical doctor, but focus on the pathology. And he come from north part of China and very cold. And in the winter is long, and everybody is sitting in the room and smoke, second hand, the first hand, and make the smoking environment. And he want to look for that, just how that affect for the health. And I said, I told him, you come to the good place because tobacco and this is the hometown, and we can work on the cadmium and how the low dose cadmium level to affect the health. And so that is, we gave him this project, and we start working on that. So when we look for the literature, and we found if you look for the acute dose and the high dose, that already have a lot of publication affect all the organs, because high dose cadmium acute infects. So that's nothing new. So I told him, if we want to mimic that situation, probably we need to use a very low dose and long term to look for the model. So we expose the rat with very low dose cadmiums, and that is we call the 20 nanomole, and calculate the ppm and something around about 2.25 ppm. So we use IP injection because the first study we want to give a cure dose go to the body, not just you don't know how much got in the body. We want to see if that really cause some impact or not. So we gave the 4 weeks treatment, and then we stop. We do not immediately sacrifice the rat to look for organ damage. That is a lot of publication. So we ask them, let them survive, mimic the occupational exposure. You go to some place and work for 10 years, 20 years, and bad situation, and then you stop. We want to see later what's going on. So you can see even after that, we can observe until 52 weeks after treatment. So we want to see that chronic effect. And we collect the kidney, liver, and heart, and testes. And this is the kidney and cadmium levels. You can see they have a break. And you can see normal group. And even the 4 weeks treatment, and this control will sit in, and after 20 weeks, so this control level is very low, around less than 40, around between 30 to 40. But you can see the cadmium treatment group, even 20 weeks after 4 weeks treatment, still more a tenfold high, and about 380, and compare the control. So that's more than tenfold increased. And even until the 52 weeks after treatment, still keep the kind of relative high. You can see 300 and tenfold increase, compare that. And for the kidney, not just have the cadmium accumulate there, but actually affect other essential metals. For example, zinc. You can see even the 20 weeks after treatment, zinc level significantly lower than control. We know how important zinc in our body. So that is until very late and time points catch up to the control level. So that is one bad phenomenon. And for the copper, and copper is a very reactive metal, and that if too much is not that good. You can see and make the copper increase compare the control persistent increase. But calcium at the beginning and 20 weeks after treatment, early stage, still low, later catch up. So that causes the dyshomeostasis of the essential metals. We look for the kidney functions. We found that not that much change, and look for the BUN only one time point and increased, and the temper and go back. But for the creatinine concentration and very little increase, also around 36 weeks and significant difference, but later is very close. Basically, not that much affect the kidney functions. But look for the detail, the morphology, and because of his pathology, he interested in the pathology and structure, and look for the different time point, and you can see and 20 weeks control ketamine and 28, 36, and gradually see more some damage, and the tubule have some disarrangement, have some the very dark pink cells, and he said that is a kind of the dying cells from the tubule. So that is a long, persistent, mild damage and cell death, and look for the a glomeruli and have some accumulation for the collagen. And so you can in glomeruli and the significantly persistent increased and at high level. And look for the biochemical assay and from the kidney, and we found not that much change. You see GSH, we know that is one antioxidant, and MDA is a kind of the damage, not that much increased, but only SOD, you can see in the late stage, significantly the ketamine treatment significantly lower than control levels. You can see from the three group. So that means the SOD and one antioxidant decreased. We know methylation, if you're treated with a metal, and that is a very important proteins. And this is the control. You can see that is a age-dependent increase until very late stage. But in the ketamine treated group, age-dependent decreased. The longer the expression lower. So that means, and that is an antioxidant function, SOD and methylation is decreased. And we look for the cell proliferation, and from glomeruli, and you can see some PCA and increased, and there's a proliferate. And at the beginning lower, but later and catch up and over the normal levels. And from the tubule, same thing, and that and increased and the later stage at the beginning and get shut down, but at the later stage and proliferate. So that is what we learned from the renal ketamine toxicity. And how about the livers? So still, Dr. Brown, and he collect the liver and first and look for the liver ketamine concentration. And 20 weeks after treatment, and still high like a kidney, but not that high like a kidney. That 300, 400, 300, and that is that level. But here you can see just fourfold, and but later almost close, but still a little bit high in the very late stage. And so that indicates how stubborn and difficult to remove and ketamine from our body. And so zinc and relative early catch-up control level. So something is better than kidney. And so look for the morphology, not that much, and you can see only here and some around the central, and you see that some damage and inflammation. And if we look for the oxidized lipid, and in the control not that much, but the ketamine group have a significant increase, persistent increase. So that is a oxidized lipid and the MDA and higher than control, and from 20 weeks to 42 weeks. And look for the cell proliferation, like a kidney, and we use the two markers and the PCA, and you can see the ketamine group, a relative high compared to control, even at the beginning not that much, at a significant and even less. You can see quantity like this, but later increased. And if we quantity for the Ki67, that usually we stain for like tumors tissue to look for proliferation. You can see this group and significantly higher than control. So, and we're using the proteomics and to look for the mechanism versus like a molecular level change. And we use like a 2D gel, and you can see there's a lot of the dots. But we are interested in two example, and like number 7, that DDT, and that is a 25, is that the Ph.D. that are prohibiting. And you can see the number 7 control, they have like that dot. But in the here is gone. And this one also like a 25 is here, but here is much less. So quantity that both significant decrease by ketamine group. And we're using Western to confirm Western blot in the whole liver, and we can see that the DDT and like a time-dependent decreased significantly, and the PHB is same thing as like a protein omics, and like a DDT and the PHB and decreased. And he also do staining because that melanin is a kind of the antioxidant, but the melanin is one end product for the DTT, and so he did staining in the control, have a strong and melanin existed there, but not ketamine treatment group. But that reduced, the activity reduced, and cannot generate more the melanin in the livers. So that is some the protein part that we look for. And we look for the antioxidant. In liver is have a more antioxidant decrease, SOD and also GSH, not like a kidney, only SOD. And when we look for the MT staining, and you can see control that age dependently increased, but this one decreased. So what we learned from the liver, and we found that the chronic exposure to the low-dose ketamine in vivo and make the liver show some long-lasting isogenic change. And this change probably due to the persistent inhibition the antioxidant function, such as PHB or methylcyanin. And we think this is the chronic long-term and have like a chronic cell proliferation, probably some risk for the cancer and the development. So he continue and addresses the question. And first, he wants to see if that ketamine really cause some genomic damage or not. So use the GABA and HAX, that is one measurement for the double-strand DNA break. And that is the red color, and this is acting and showing the cell's structure and the kind of graph, like a picture. And you can see this is a lone staining. If overlap, merged together, you can see the green background, but a lot of the red dots and red nucleus. When we enlarge that, we can see that nuclei have a lot of the foci. So that is a kind of DNA damage increased. And usually we know our body have a clear and very strong and apoptotic cell death pathway. If any cells have some bad genomic material and they don't let your keep growing and turn on the program and let your apoptotic cell death to remove the bad cells. So we look for the tunnel. We found in the ketamine treatment group at the later stage, and tunnel cells, positive cells, is lower. We look for the cleavage, the Caspar-3, and same thing, and that is decreased. So when we look for the cell death pathway, we know they have a multiple pathway, mitochondrial pathway, ER stress pathway, and like some mechanism pathway. But we also know that like a TNF-alpha, phospholigand, the receptor pathway. So basically like a TNF-alpha there, and turn on the Caspar-8 and make the Caspar-8 cleavage, and that turn on the Caspar-3 and cause the apoptosis like this pathway. So we want to trace that pathway. We look for the cleavage of Caspar-8, and we see and that is like the ketamine group. Then like this kind of is less compared to multiple error here, and we look for and TNF-alpha staining and the ketamine treatment group increased. So we want to see how come, and that is a kind of make the TNF-alpha mediated apoptotic cell pathway kind of broke. Top is increased, but the bottom cannot match up. So we know and cadmium can induce a lot of the epigenetic change and predominantly cause the demethylation and abnormality. So we use the demethylation array and to look for the control and the cadmium treatment group. We found cadmium group and treated and turn on a lot of the methylation and also down some promoted the methylation. And because we are specifically interested in TNF-alpha and caspase-8, and so what we found, and caspase-8 gene promoter and got the increased methylation, and TNF-alpha promoter and got lower methylation. So what come out, and we think from low dose cadmium and make the promoter of the TNF-alpha and hypo, then reduce the methylation. So that means that promoter still can efficiently affect by transcription factor turn on the TNF-alpha, keep the increased. And for the low dose cadmium, probably higher the methylation for the caspase-8 and the promoter. So that kind of shut down the transcription factor to binding to turn on. So that makes the caspase-3 total expression reduced. And so that makes the pathway broke down. Top still keep increased, but the downstream cannot respond. So that is make the cells have more DNA damage, but have less apoptotic cells as a mechanism. So that probably is a risk for cancers. To want to confirm that, and the hypothesis is correct or not, we set up the mouse model, again, give control cadmium treatment. But we also gave the cadmium treatment with the methylation inhibitor. This is the long name, and the short name is 5-AZA. And we also have the control with the AZA, and we treated as 4 weeks and let them survive until 56 weeks after the treatment. So totally 60 weeks. And first we look for the caspase-3 and the promoter methylation. You can see cadmium significantly increase that methylation. But if we have a methylation inhibitor, of course, that even lower control because we have an inhibitor. And meanwhile, but if we have cadmium treatment, we also with the methylation inhibitor that shut down that. So we can see the caspase-3 cleavage caspase-3 and cadmium treatment still low. That is consistent with the previous study. And if we give the a methylation inhibitor, that kind of reduce normal level to a little bit, but revise that to the cleavage caspase-3. So that is a same model that clearly and explain that. So when we go back to see the apoptosis and tunnel and the CBC, cadmium treatment reduce tunnel, but inhibitor methylation can prevent cadmium inhibit reduce the tunnel positive cells. And cleavage caspase-3 is same pattern and consistent with that. When we look for the cell proliferation, we use a Ki-67. You can see cadmium treatment group increase that. And the Western blot also confirms that, and that increases the cell proliferation. And if you have a methylation inhibitor, can prevent cadmium increase the cell proliferation. So that is a perfect explain and what we found. So we want to see if, okay, do this liver have some marker or early stage phenotype of the cancer? And we look for the morphology. They have a kind of the cumulative, a vacuum and a bad liver cells and have inflammation cells. And when we look for TGF beta, that is one kind of involves some carcinogenesis step. You can see cadmium increase that. And we also check with the CK8 and the 18. They have a cocktail antibody, a cocktail. They can detect both, and 8 and 18. And so you can see the two band. Cadmium increase that, and but methylation inhibitor decrease that. This is a kind of cytokeratin and 8 and 18. That is a neoplastic and the markers. So you can see, and cadmium increase that, and if we have methylation inhibitor, reduce that. And when we do the staining, you can see that. So this is the control, and so this is the HE staining, this is a CK8 and 18, so that's control and like a methylation inhibitor, much less compare this. And the ketamine with methylation inhibitor, not that much different from here. But if only ketamine alone, I pick up different field, you can see that kind of regional have different kind of dark and mild dark and like a positive staining for the CK. So that is kind of early proliferation and early neoplastic phenotype. So we learn under the chronic and the exposure to low dose ketamine and in vivo cause the liver have methylation change, increased, decreased, and that probably causes the persistent cell proliferation, and that is the methylation and causes the apoptotic cell death pathway shut down and not parallel, and one in TNF-alpha increased, but Caspase-8 pathway got stopped. So that is makes the less apoptosis and increase the cell numbers. So that is like some hepatic toxicity, and we also like look for the cardiac toxicity and the test toxicity. So I won't share the slide experiment date, but I can summarize that. What we learned in the kidney, and we found long-term and mild injury and proliferate cells and decrease the antioxidant system. In the liver, we also found oxidative stress and damage and reduce antioxidant, less apoptotic cell death, and hyper and hypo demethylation, and like a lot number change. And the chronic inflammation, and also with the cell increase cell proliferation, and how early and like a neoplastic the marker increased. So that probably kind of is some cancer symptom on like a warning. So in the heart, and we found hypomethylation and increased, and the myocyte contractibility decreased, and have some mild fibrosis. And if we give the inhibitor of the methylation, and that can restore cardiac function and reduce the fibrosis. In the testes, we found some damage, and more important, we found that the DNMT1 and the DNMT3A, that is both is different kind of the demethylation enzyme. That not change, only B, and the B usually normally in the testis not active. That ketamine turn on the novel and methylation enzyme, and probably that is the one reason cause the more hypermethylation. And they have a DNA damage, and they also have a cell proliferation, and that is a kind. And if we inhibit the methylation, and can prevent that effect. So that is the first model. We just expose the adults and mimic the occupational, and then look for your long term. But now we want to see a lot of people just leave the environment from the embryo, in the body of the mom, and start the mom drinking like water or smoke, and already affected the baby until they are like independently after weaning and independently survive and adulthood, what's going on for the whole life exposure. And so that is a second models. Because that is probably also how more people population involved, not like occupational only a few, and if not dangerous, you can away from there and easier to avoid. But the whole life exposure not that easy. So this is the model, and we think probably more mimic a human situation. And actually, like I mentioned, you have one, and probably also have a number two got the challenge and the multiple factors. So because we work on the like diabetes and obesity, so we pick up the obesity as the second hit for this model. So this give you a cartoon explain. So that is like and when we got the mice and mom got the drinking water and start to use to the environment, and then give the drinking water include ketamine and have low dose and 0.5 and high dose 5 ppm, and then let them like 2 weeks drinking the ketamine drinking water, and then breeding with like a male, and got the pregnant and start that, and during the whole embryo development stage, and until the after birth and until weaning, so that the baby, the offspring, got the ketamine through mom drinking water or placenta to affect the individuals. Once they got weaning, and this mice still keep the pattern what parents give them normal drinking water, and just still continue normal water. They have ketamine water and ketamine water, but both have a low-fat diet and have a high-fat diet. Second challenge, mimic the obese children, and they are not pay attention, eat too much and overweight and overnutrition, and how to see that is make the individual more sensitive or resistant. So that is our goal. So Dr. Zhou and work on the male reproductive system, and Dr. Humbo men and work on the liver and look for the IGCC, and Dr. Jim Yang and as a project leader work on the systemic and also work on liver and NAFLD, and some other people work on the heart and kidney and the brain and the lung. So we have a teamwork and work on the different organs to see what's going on there. So this is from the Jim Yang state, and we first want to see is a weaning, after weaning, post-weaning hepatitis really cause the obese and have some overweight. And from this dynamic analysis, you can see that hepatitis really increase the body weight, got obese. And is the ketamine also affect, cause the obesity? And actually, in the normal diet, ketamine, no matter high dose and low dose, they are not affect. The body weight basically same with the normal diet without any ketamine. But in the high-fat diet induced the obese, but the ketamine have some impact. Low dose ketamine reduce the obese in the later stage, but high dose ketamine increase it worse. So that is the dose dependent. And this is the accumulation of the ketamine in the liver. You can see normal diet, dose dependent, and the high-fat diet, and still dose dependent, but compare this difference. Low dose, higher, more ketamine in the liver, and the high dose group not that much increased, even a little bit reduced, but still very high. So this is some difference and dose dependent. So this work already submitted the journal Toxicology and Applied Pharmacology, and they allow us to revise. We plan to submit back in January, and basically it's a minor revision. And this is a graph, the abstract. So this is our hypothesis, how ketamine plus high-fat diet in the post-weaning, 1 + 1 = 2, or = 3, or = 1.5. So this is our questions. So this is how to treat the animals. As you can see, the parents, and they got drinking, and with without ketamine, and have a high-fat diet or not for the upstream. And then we look for the liver, and you can see we just shown the high-fat diet and with the ketamine group. You can see high-fat diet in the control without ketamine treatment can really increase the lipid accumulation in the livers. And this HE staining, this oil red staining is a lipid, red is a lipid and staining. And low ketamine treatment makes that less and reduce that phenotype. But high dose ketamine worse compared to the without ketamine. You can see that pattern. So this is like a systemic and observation for that part. And we also submitted the heart and work and to this journal, and actually that already accepted online available. But eventually the exact page with the issue probably next year. Right now it's December. So same model, but we will see, and we look for the heart and the cardiac function, and we found the fibrosis and the hypertrophy and the remodeling, and we look for the ketamine accumulation, and the trace elements caused change, reduced, and RS increased, antioxidant decreased. That caused some damage, but that predominantly in the female, it's not a male. So that is something is unsurprised, very surprised. So this is the example, and when staining calculated the cell how large based on the mark, and this is a normal without ketamine, and this is the normal diet, high-fat diet. High-fat diet alone can make the heart become hypertrophy. That we know already, and obese can cause cardiac function in the long term. And if we have a ketamine, that is the worst, synergic and increase that hypertrophy, or the other measurement from different level, protein level, gene level, show same patterns. So kidney, and we see that we want to see is ketamine alone, and you see, compare the control, low dose ketamine didn't change that much, even reduce a little bit protein in the urine. But high dose ketamine and increase that a little bit. And other measurement, ketamine didn't induce that much. But you can see if we have a high-fat diet and the control and without ketamine, you can see high-fat diet increases the protein urine like that. We know that causes cardiac dysfunction, causes kidney dysfunction. But when we give a low dose ketamine, reduced. High dose ketamine, worse that. And so other measurement, the same pattern. And when we look for the fibrosis and in the glomeruli and the same thing, and the low dose ketamine and have a little bit better, and but this high dose ketamine is totally worse. So that is in the kidney part. We haven't published yet, so we work on that and work with Dr. Madhuri Rin, and know her. And so now I want to go to the cancer part. And this cancer part, we want to see how the ketamine affect the individual sensitivity. So after the weaning, we didn't continue to give the ketamine treatment. We just gave the cancer-induced diet to see how the sensitivity changed. So this is the model, and parents and got ketamine treatment, and once under the weaning, and we gave the cancer diet called HFCD, that high-fat diet with the choline and deficiency. That is one commercial available induced HCC models. So we want to see how the ketamine affect that second his. And after weaning, we don't have a ketamine, but until 6 months. And the body weight, and you can see that is mom do not have drinking ketamine, just pure control. And this is the offspring gave the HCC diet to induce cancer, and this female, this male, and this one, before weaning got from mom got drinking water, called some ketamine treatment, plus the HCC induced cancer. So that is the two factor. So you can see both group and increase the body weight and there. So, but bottom line, we want to see cancer, that body weight just one indirect fact. So this is the tumor numbers, and we at the 23 weeks and not last in the middle, and we sacrifice 3 just individuals, control, of course, not liver cancer, and cancer induced die, induce 2, and that liver cancer. But if they have a pre-ketamine treatment, all the 3 can become cancers. So that is a significant difference in the male, but not in female. And you can see that is different. And this is the size, and this is the liver nodule. And when we look for the liver and to the TB length, the ratio, you can do how to compare how large the livers. You can see in the female, not a big difference, but in the male, they have the dependent increased. Cancer inducer plus ketamine, and significant increase compared to this control. And not just the number increase in the male, but in each nodule, the size. And if we decide a subgroup, that also is significantly higher than the cancer inducer only. So that means ketamine plus HCC inducer worse and induce the cancers. And this is the HE staining nodule. You can see that a lot of the lipid and the position and the accumulation. And if AFP, that is a cancer as a marker, and it's a significantly increase that. And the mechanism we think is and the lipid metabolism abnormal, and it got more fatty acid go to the cells, but that is kind of stop the mitochondrial beta oxidation, so cannot use efficiently, so got the accumulation and cause lipotoxicity. So that is published already. And what we learned from this is like a two model study. We learned the whole life exposure to low dose ketamine not directly causes significant damage for multiple organ, not that much, but a little might. But that is the change and the chronic inflammation or somehow probably enhance the second chain, like a hepatite causes the phenotype, and further increase the sensitivity. But that is dependent on the dose. Low dose, not. High dose, increased. So what is the futures? And we want to know what is the mechanism. Why that enhance that sensitivity? And if we found the mechanism, probably we can apply some approach to intervention. And also we want to know why small dose not to enhance that damage, even protect. And is that protect have some really beneficial for our health? Like apoptosis, cell death, if you are reduced, it's not good. So we don't know what is the long term, what is the final outcome. And just to see the toxic effect is reduced, but we don't know, probably more cancer in the future. So now I want to start here, but I appreciate all the work and it's done by my lab member, and particular special appreciation to Dr. Jimmy Yang, and she is the leader for the project. And I also want to thank the long-term collaborator, Dr. Yitan, Copper Wintergers, and Jun Kai in there, and also other group like Dr. John Weiss and Chris Dade and John Friedman and somebody others. And this is a list of some foundation to support, not exactly this project, but it's my lab. So, and thank you very much for your attention, and I'd like to answer if you have questions, but don't forget the number you need. Thank you.
Sharing.
So, yeah. Is this one? This one, right? I think I have two. Thanks, Dr. Tsai. How does one measure cadmium in the environmental or body fluid? So we and our Pediatric Research Institute, we have an IPC mass that is can measure and some material from the water and from the tissue, and as long as you can use an acid, nitric acid, and digest, and we can measure even bone, and we can measure that the cadmium level from there. And that machine can give you multiple. That's why you can see we see the cadmium, also see the other metal change, and they can show about 20 or even more, and the metal come out, calcium, potassium, and copper, and so that is a very powerful machine. We have that. Yeah. Thank you. And another question. Yeah, that's only one question.
So is the in Turkey?
Hi, yeah. But right now I collaborate with Dr. Charlie Zhang, and he work on the the geography and try to identify which location they have a map. And we want to apply some small project to see if that really link the prevalence or not. But from the EPA data, looks like Kentucky is high. Yeah.
So does cadmium bind to adipose tissue?
Adipose tissue? Yeah, and that cadmium they call it mitoestrogen, and kind of have some estrogen functions. Make the insulin resistant and cause chronic inflammation and stimulate the adipogenesis. I think so. The distribution, fat and the muscle and the kidney, liver, but from the preferable to go to the bone because it replaces calcium. So that is a target organ, and other organ is the liver first. But the liver has a kind of powerful mechanism to methylation and remove all the cadmium go to kidney. Kidney is not that lucky, not that much protection, and slowly release the cadmium, and the cadmium release, and during the elimination cause some damage for kidney. So that's why you can see liver quickly go to lower, but kidney still very high and tenfold, more than tenfold. So we didn't measure this model for the adipose tissue, but that is the whole body distributed part. and kidney and liver and also brain and increase.
So is there something we can do to get rid of cadmium?
We use zinc induced methylcyanine at least can binding and can prevent your acute damage, and then slowly to remove even kidney probably cause some damage. But more zinc is better induced and methylcyanine, and zinc have a binding facility much less, and copper have a 10-fold, 20-fold power to binding methylcyanine. So that is one way you use.
So the zinc deficient cadmium toxicity.
Yeah. So that is one way is right now to use zinc to induce the methylcyanine. Don't let the cadmium-induced methylcyanine binding strong and damage. Yeah, so can't replace.
More questions in the chat.
Over. Have anyone mapped cadmium around the Louisville? And I need to ask Dr. Charlie Zhang. He right now is a calculate and EPA date, and they have the date and multiple metal, but didn't come out yet. That is our interesting question too. Does cadmium stay in the elemental form of the compound in the body? So what means elemental? You mean that in the free cadmium?
Cadmium, just in the elemental form.
Yeah, usually cadmium is binding with hemoglobin and most time binding methylcyanine. And so that is they have one technique use hemoglobin binding and to cadmium assay to measure methylcyanine, and they use cadmium to replace to calculation for that. Yeah.
So it's bound to some to protein, either hemoglobin or metallothionein.
Methylcyanine predominant.
Thank you.
Thank you. Yeah. Thank you very much for your attention.